gene, right, we saw that you have you have two homologous, or you have a homologous pair, right, two homologous chromosomes, and on them you are going to have alleles, but they're the same gene, right? So two copies of each gene or alleles, and these are separated and equally likely to be inherited, right? So when I go through meiosis, all this means segregated means separated, and when they're separated and eventually, right? So let's say I draw the cell, and I have chromosomes in them, right? Chromosomes. Eventually, those chromosomes are going to get split apart, right? They're going to get pulled apart from each other, and eventually, I'll get I'll get a bunch of cells, and they're all going to be different, right? But the odds that any of them will get split are equally likely, right? There's two possible options. That's all that's saying. Okay, let's keep moving. And another law is the law of dominance, and we talked about dominance already. So the law of dominance. Oops. Dang it. There we go. Law of dominance. So each organism contains two copies of genetic information. When two copies of the particular gene don't agree, the dominant allele determines the trait, right? So we saw that in those different colors. So if we had big Y and we had little y, even if I have a little y here, this big Y is dominant over it, and it resulted in a yellow pea pod. And so when dominated, the recessive allele has no effect, right? This little tiny little recessive, little tiny recessive allele had no effect. Okay, easy enough. You already knew that. And we have laws of independent assortments. We already talked about independent assortments, right? So when they're split in meiosis, they can have a 50-50 shot of being split evenly, right? Or they can line up in a certain way, and that results, right? You could take, I could go there's two possible combinations, and there's 23 sets of chromosomes, so 2 to the 23rd power is roughly 8.4 million. And those are my odds of them dividing in such a way that, or aligning in a way, so they can align during gamete formation. An example is creating a gamete with brown hair and freckles is equally as likely as creating a gamete with brown hair and no freckles. So they do not determine each other, right? They're independent of each other. We will see later that there are some traits that can actually be linked on a chromosome. So an example of that would be like for people that have red hair, right? So usually if you're, I guess what people call if you're ginger, or if you have red hair, often another trait that you get with that is freckles. It's more, and another trait is it's even more rare to get like blue eyes and have red hair. Because oftentimes those are actually linked, and we'll talk about sex-linked traits in another video. But okay. So you can see here, this is another video that you can watch, and this just goes into more detail on Gregor Mendel. I think the Edpuzzle videos cover it well enough where you could probably skip this video. So let's get into some problems. So how do we solve some basic genetic problems? Okay, so back to this picture. So how will a trait be inherited? Well, it depends, right? You can have the way Mendel said it, and what people found out later is there's actually other more complicated ways that things are inherited. So non-Mendelian. You can have dominance. You can have what's called incomplete dominance or codominance. So sometimes it's actually true that you'll see a blending of traits. There's not necessarily one that's dominant over the other one. And you can have more than 2 alleles, right? There can be more than one gene. It can be sex linked. It can be autosomal. Your environment could impact it. So there's a lot of different things here. But we're going to focus here today on Mendelian genetics. So basics is that controlled by one gene, 2 alleles, and either there's dominance or recessive. An example of this is hitchhiker's thumb. So in humans, hitchhiker's thumb is dominant, and so Mr. S, as an example, has hitchhiker's thumb. Let's assume his genotype is TT. Now, what do we call this? Let's see. Do we call this homozygous or heterozygous? I'll give you guys a second to think about that. So if you took a second, yes, you're right. This is, let's write this in. So this is heterozygous. Heterozygous, right? Hetero because they're different, big T, little t. His wife, however, has straight thumbs. Right? You can see this image. Nice and straight. Doesn't bend backwards when you give your thumb. So see, even I have the hitchhiker's thumb. When I put my thumb up, it bends backwards versus her thumb stays nice and straight when she bends it. What is the probability that our future children, or so his future children, will be born with hitchhiker's thumb? Let's try this. So what we're gonna do is create a Punnett square. So where do I want to make this? Let's make it right here. And Punnett square, all we do is just make a 4x4 grid. And let's see, well, we have to, let's say on the top here, we're going to do mom. And here on the side, we're going to do dad. And let's make the possible gametes. Well, we know his genotype, Mr. Cervantes' genotype is TT. So let's put that in. So his is big T, little t. Right? His wife has straight thumbs. So what does this mean? Well, let's read this question again. So hitchhiker's thumb. So when your thumb bends backwards, that is dominant. So let's draw out the possible allele combinations. So let's draw out the possible genotypes. Genotypes. So our possible genotypes, we could have, there's 3. Let's see if you can do them before I do, but you could have big T, you could have big T, or you could have little t, right? Okay, so let's see. If I had big T, this trait is dominant. So if I have big T, then that means I'm going to have hitchhiker's thumb. If I have big T, I'm still going to have hitchhiker's thumb because it's dominant, right? But if I have little t, this is recessive. Now I'm going to have a straight thumb. Straight thumb, yay. Okay, so if his wife has straight thumbs, what's the only possible genotype she can have? That's right, she can only have little t. So let's do our combination. So all you do is we're going to start here, and we'll cross it up here. So let's imagine we put these two together. So I could have one set of gametes that has big T, little t, or this is offspring, sorry. So this is, I could have offspring that have this. Now let's cross this big T with this little t. I could still have big T, little t. And let me get my eraser, get rid of those. Now, what if I crossed his other gamete, right? And I crossed it up here, I could have little t. And if I did the same thing with this one, it would be little t. So now, what's the probability? The question is, what's the probability that our future children will be born with hitchhiker's thumb? Well, let's look. So we have our key over here. Which genotypes result in hitchhiker's thumb? So it looks like only these two that are circled result in hitchhiker's thumb. So let's see. So out of 4, it looks like 2, and I'm going to say have hitchhiker's thumb. And the other one is that it looks like there'll be 2 that have straight thumbs. So what's the probability? Well, my probability, you could just say 2 to 2 ratio is the same as 1 to 1, right? Another way to say that is just that it's 50%. There's a 50% chance, right? Because half of the time I'm going to have no or straight thumbs, and half of the time I'm going to have hitchhiker's thumb. Okay, let's see. It's clear. So here's kind of some general steps to solving this. Is you want to read the problem carefully. Underline important words like hybrid, heterozygous, or dominant. And you saw me just instinctively do that, but you want to say, okay, what is the trait, or what is the question asking? And the question is asking, let's do this here. What is the probability that our future children will have hitchhiker's thumb? And what you want to do is then look at, well, which trait or which trait is dominant, and we saw that hitchhiker's thumb is dominant, or you want to see which one is recessive. And so we did that, and then we said, okay, what is the genotype that one of the parents, at least one of the parents, has, and what trait does the other parent have? In this case, straight thumb. And then you want to draw those allele combinations. So let's not get ahead of ourselves. So right, dominant alleles, capital letters, recessive alleles, lowercase letters. You want to write out the genotypes of each parent, right? So it might help to actually write out all the possible, it might help to actually write out all the possible genotypes, right? Big T, big T, big T, little t, little t, little t, right? Drawing out all those combinations, and then drawing your Punnett square. And we say, okay, I know that the mom is this one, or I know the mom is this one, and then lining those up in the Punnett square and saying, okay, mom's up here, dad's over here. Uh-oh. It's not wanting to cooperate on this side of my screen. Anyway. Mom's there, dad's here, and lining up those corresponding genotypes and phenotypes. Okay, cool. Let's clear that. Let's keep moving. So here's an example of something similar we did. So they could also say that it's a cross between a homozygous dominant or a heterozygous dominant, or you can see they can just use the words, right? And you have to follow along with that. So homozygous, right, meaning same, but they're recessive. So I know the little letters have to go there. And I know this one is homozygous but dominant, so I know it has to be both big letters there. Let's do another one. So green leaf lubo plant, I made this plant up, yeah. Has yellow striped leaf. This cross produces 185 green leaf lubo plants. Summarize the genotypes, phenotypes of the offspring that we produced by crossing 2 of the green leaf luba plants from the initial parent plants. Okay, so they made a green leaf luba plant. So let's figure out what we're trying to do. So, okay, again, so let's read exactly what this problem is saying. So it looks like our, like this, white, so our, they crossed a green leafed plant with a yellow striped plant. And when they did that, they got only green leaves. So we took a green, we crossed it with a yellow. And so this is our, we'll say this is our P generation. Dang it, it does this. This is our P generation. And then our F1 generation, so the one below this after we crossed, only resulted in green. Okay, so that's what we know so far. Now we want to know, so what if we took those offspring, so we took those F1 generation and we crossed them with each other. So now let's say we cross this green with this same F1 generation. What do we expect for our genotypes and phenotypes in our F2 generation? Well, let's think. So what do we know about dominant and recessive? So from our parent generation, which of these is dominant? I'll give you guys a second to think about that. So you might have paused and thought about it. So green is actually going to be our dominant. Why? Well, because I crossed a green and a yellow and I still only got green, right? So I know green is dominant. And that means that Y has to be recessive. So if I know green's dominant, Y is recessive. Let's see if we can make phenotypes for this. So I'm just going to say that since we're talking about leaves here, I'm going to say that L stands for leaf color. And I'll say big L is for green. Or you know what? I'm going to make these not colored. So we'll say that big L is for green. And we'll say that, so maybe we'll do this here. How do I want to do this? So we're going to say big L is for green, little l is for yellow. So now if I make all my allele frequencies, I could possibly have big L, big L, little l, and I could have little l, right? What do I expect from these? Well, I expect if anytime I have a big L, right, because it's dominant, I should have a green. Anytime I have a big L, I should still have a green, but now I have two little L's, so now I'm going to actually expect yellow. So what does that mean for my F2 generation? Well, if I took if I made a Punnett square, let's figure out exactly what that would look like. So I'm going to make a Punnett square right here. And let's say we know down here in our F1 that because we, well, let's do our parent generation first. We got to do our parent generation, or sorry, F1 generation. So we're going to take our parents up here. We're going to take our parents from up here, and we know that if these were true breeding, so that means that green has to have the phenotype or genotype, big L, and my Y has to have the phenotype, little l. And so when I cross them, all of mine were green, but you can see that they're heterozygous, right? Because they still carry this yellow genotype or yellow allele, right? So now let's do my F2 generation, because that's what we're trying to figure out. We're trying to figure out our F2 generation. So now we know that all of these F1 have the genotype big L, little l. So then we know if we cross them with each other, it's just going to be big L, little l, big L, little l. And now let's actually cross those. So these are going to come together. So I have big L, big L. This big L is going to come with this little l, so big L, little l. And then now this little l down here is going to come up and cross with this one. So now I again have big L, little l. And then it's going to do that one more time with this little l, so I have little l, little l. So what do I expect from this? Well, I expect that there's going to be, what's my genotype and what's my phenotype? Well, let's focus on the genotype. We're going to expect, let's think of our ratio. Which of these, I'm going to put a circle around these, which of these are going to be green? I'll give you guys a second to think about that. So the ones that are green are, like you guessed, right? Green is dominant. So anytime I have a big L, these are going to show up as green. So I expect a ratio of 3 green. And which one's left over? Which one's yellow? Well, it's this recessive, right? Little l is the only thing that's going to get me yellow. So I expect a 3-to-1 ratio. you could actually figure out the, well, could you figure out the exact number? Only if they told you the F2, the number of F2, could you figure that out. And we'll maybe give you another question like that later. I hope that made sense. So again, you want to figure out which one's dominant, which one's recessive, figure out your possible possible allele combinations, your possible genotypes. And then you want to figure out, okay, what has to be my F2 or F1 generation? What are those phenotypes? And what is my F2 generation?